Hello friends, welcome to our YouTube channel sripedupharm. I am Mahendra Kumar Sahu, assistant professor in SRIP Kumari. So today videos we will be discuss about urea cycle. The topic is covered introduction, synthesis of urea, significance and disorder of urea cycle. Okay, so let's start. The urea cycle is the first metabolic pathway to be elucidated. Next point is the cycle is known as Krebs-Henselt urea cycle. Next, ornithine is the first member of the reaction. It is also called as ornithine cycle. Urea is synthesized in liver and transported to kidney for excretion in urine. The two nitrogen atoms of urea are derived from two different sources, one from ammonia and other directly from the alpha amino group of aspartic acid. Okay, the next is carbon atom is supplied by CO2. This CO2 हमको मिलता है हमारे TC cycles. Okay, next is urea is the end product of protein metabolism. आप कह सकते हैं amino acid metabolism. About 80 to 90% of nitrogen containing substances excreted in urine. Okay, next point is urea synthesis is a 5 cyclic process with 5 distinct enzymes are involved. Okay, then next is the first two enzymes are present in mitochondria, will be the rest in cytosol. Okay, this is the overall cycle of urea cycle. So let's start this 5 step to step by step. Okay, so let's start. First step, formation of carbamoyl phosphate. Okay, the first point is carbamoyl phosphate synthase is an enzyme present in a mitochondria catalyze ammonia ion with CO2 to form carbamoyl phosphate. Okay, the next step is this step consume 2 ATP and is irreversible process. Okay, this step is a rate limiting step. CPS first are carbamoyl phosphate synthase in first enzyme required N-acetyl glutamate for its activity. This N-acetyl glutamate are cofactor या coenzyme है. Okay, the next is carbamoyl phosphate synthase second involved in pyrimidine synthesis and it is present in cytosol. Okay, the next it accepts amino group from glutamine and does not require an acetyl glutamate for its activity in case of carbamoyl phosphate synthase second. Okay, CPS first are carbamoyl phosphate synthase in first enzyme required N-acetyl glutamate for its activity. This N-acetyl glutamate are cofactor या coenzyme है. Okay, the next is carbamoyl phosphate synthase second involved in pyrimidine synthesis and it is present in cytosol. Okay, the next it accepts amino group from glutamine and does not require an acetyl glutamate for its activity in case of carbamoyl phosphate synthase second. Okay, second step is formation of citrulline. Okay, the first point, the second reaction is occur in a mitochondria. Okay, next is citrulline is synthesized from carbamoyl phosphate and ornithine by ornithine transcarbamylase enzyme. Okay, ornithine is regenerated and used in urea cycle. Next is ornithine and citrulline are basic amino acid never found in a protein structure due to lack of codon. Okay, next is citrulline is transported to cytosol by a transporter system. एक ट्रांसपोर्टर सिस्टम के थ्रू वो माइटोकॉन्ड्रिया से साइटोसॉल में ट्रांसपोर्ट होता है. ओके, नेक्स्ट इज citrulline is neither present in a tissue protein in blood but it is present in a milk. Step fourth is formation of arginine. Okay, in this reaction the enzyme are involved arginosuccinase or arginosuccinate lyase. Okay, this enzyme are cleavage arginosuccinate to form arginine or fumarate. Fumarate is an intermediate product of TCA cycle. Okay, then next is fumarate provide connecting link with TCA cycle or gluconeogenesis. Okay, step 5 formation of urea. Okay, first arginase is the fifth and final enzyme that cleaves arginine to form urea and ornithine. Okay, second, ornithine is regenerated, enters into mitochondria for its reuse in the urea cycle. Okay, next is arginase is activated by cobalt and manganese as a cofactor या फिर आप कह सकते हैं coenzyme. Okay, next is ornithine and lysine compete with arginine, for it is an example of competitive inhibitor. Arginase is mostly found in the liver, while the rest of the four enzymes of urea cycle are also present in other tissues. Okay, next, arginine synthesis may occur to varying degree in many tissues. Okay, the next point is but only the liver can ultimately produce urea. Okay, ओके आप लोग यहाँ पे देख रहे होंगे ओवरऑल यूरिया साइकिल. ठीक है, तो जब फर्स्ट पॉइंट था आपका CO2 combine with ammonia to form carbamoyl phosphate with the presence of carbamoyl phosphate synthase first. Okay, this enzyme required N-acetyl glutamate or two molecule of ATP to form carbamoyl phosphate. This carbamoyl phosphate combined with ornithine and transported mitochondria to cytosol with the help of transporter to form citrulline. Okay, citrulline is converted into arginosuccinate with the presence of arginosuccinate synthase enzyme. Okay, this arginosuccinate are converted into fumarate and arginine with the help of arginosuccinase enzyme. Okay, these fumarase are intermediate product of TCA cycle. Okay, then arginine are converted into urea and ornithine with the presence of arginase enzyme. This ornithine are regenerated into mitochondria for further use. Okay, next is significance of urea cycle. First point, toxic ammonia is converted into non-toxic urea. Next, synthesis of semi-essential amino acid like arginine. Next, ornithine is a precursor of proline and polyamines. Next, polyamines are include peptidine, pyrimidine and permine. Okay, next is polyamines have diverse role in cell growth and proliferation. Next is disorder of urea cycle. First point, the main function of urea cycle is to remove toxic ammonia from blood as urea. Okay, then next, defect in the metabolism of urea या फिर बोल सकते हैं आप ammonia to urea that lead to hyperammonemia and ammonia intoxication can be produces. Okay.